Hey, welcome to 10th Standard Science online class. Dear children, in the previous class, we learned about the different kinds of mode of nutrition and then how does the process of nutrition will be held in plants and then what are the steps that is involved in this process. So these are the concepts that we have discussed in a previous class. So in that, uh, the steps of photosynthesis, so that is absorption of sunlight from atmosphere by the help of chlorophyll and light energy conversion into chemical energy and reduction of carbon dioxide into carbohydrates. These are the processes we are discussed in the last class. Dear children, in this session also we are going to discuss about the more information regarding the nutrition process. I hope you understand the previous video. Yes, children. So now we are going to discuss about the more information regarding how does the process of exchange of gases will be takes place in the plant. See here, there is a special tissue or there is a special cells that we are going to identify in the plant leaf. These cells are only the responsible for exchange of carbon dioxide and oxygen and as well as wastage of excess water that is absorbed by the plants in the form of water vapors. So these cells what we call it as children. Yes, these cells we call it as stomata. See here, the stomata are the tiny pores present in the leaves. So, by the help of these stomata only, the exchange of gases will be takes place. So here, how the structure of the stomata that we are going to identify here. See children, each stomata has a pair of guard cell which controls the opening and closing of stomatal pore. So, in the picture that you are going to identify here, just observe it carefully. So it is in a stomata. In that, what are the green colored absorptions that you are observing here structures, that is we call it as guard cells. So what is the role of these guard cells? So after the guard cells that is followed by there is a one pore hole, right now. So that pore what we call it as stomatal pore, P-O-R-E. So in that stomatal pore only, the process of exchange of gases will be takes place. So after that, that stomatal pore that is followed by the guard cells. So these guard cells will be play a major role in the opening and closing of stomatal pore. So when particularly in the daytime, this opening and closing of guard cells will be takes place. How this process of opening and closing will be takes place? So when the water enter to the guard cells, it will be swells. Then the pore become open. When the guard cells loses the water, it shrinks and then the stomatal pore become closes. See here in this picture that you are observing in the below here, just observe it carefully. Yes, children, in the third diagram that you are going to observe here, just observe it carefully. In the second picture of the stomatal pore that you are going to identify here, what I was zooming in that pictures, see here, so that H2O is present in this here. In the guard cells, that H2O, which is nothing but the water accumulation that will be takes place. So because of that H2O accumulation, there is a pressure that is exerted in the guard cells. As a result of that, the stomata become opens. During the daytime, the process of evaporation will be begins. At the time only, the guard cells opening will be takes place because of evaporation. So after the daytime, after the completion of the day duration, the evaporation become reduces, keep on, time will be move on. Hence, the pressure whatever that is exerted in the guard cell, that has been reduced. As a result of that, that will become as a strinkle. So as a result of that, the closing of stomata will be takes place in the plant's leaf. So like this, the opening and closing of stomata and stomatal pores will be takes place in the plants. So in the first picture, that whatever is there, so just like an greeny picture, so that is in a cross section of stomatal pore that we are observed under the microscope. It will be looks like this. So when we are going to observe this one in electronic microscope, exact structure that we are going to identify as shown in the labeled parts photographs. So this is about the stomata and the functioning of stomata. So this process will be depend completely exchange of gases and as well as the exchange of water that will be takes place based on the principle we call it as diffusion. So what do you mean by diffusion? So diffusion is nothing but the movement of the molecules from the region of higher concentration to lower concentration. So this we can say that the movement of ions from the region of higher concentration to the lower concentration. So this phenomenon that will be held by the help of stomata in a plants. Hope you understand, children.
Process of stomatal transpiration. Most of the transpiration takes place through stomata. Stomata are microscopic parts consisting of two kidney-shaped guard cells. In stomatal transpiration, water vapor moves through the stomata of leaf. Water absorbed by the roots rises through the stem and reaches the tissues of the leaves through veins. A large number of spongy mesophyll cells in the leaf have their surfaces exposed to the intercellular spaces. The surfaces of the cells give out some of the water, which forms a thin film. The water from this film evaporates, and the water vapor formed saturates in the intercellular spaces. This vapor then diffuses into the other connecting intercellular spaces and finally reaches substomatal space, from where it escapes out through the stomata. Stomata are absent in the submerged hydrophytes, that is, Hydrilla, Vallisneria. Mechanism of opening and closing of stomata. Transpiration occurs as long as the stomata are open. It stops when they are closed. Open and closed stomata. Closed stomata. Flaccid guard cell. Stoma closed. Stoma open. Turgid guard cell. Thick cell wall. Thin cell wall. The opening and closing of stomata depend upon the turgid or flaccid state of guard cells. When the guard cells are in the flaccid state, stomatal aperture closes. During daytime, as a result of photosynthesis, as the guard cells have chloroplast, concentration of carbohydrates rises, leading to osmotic uptake of water by the guard cells to swell. Since the wall of guard cells towards stomatal opening is thick, while outer convex wall is thin, inflow of water in the guard cells causes them to bulge outwards, thus widening the stomatal opening. When the osmotic pressure of the guard cells becomes lower during night, the water leaves these cells due to exosmosis and moves to the neighboring epidermal cells, having cell sap of higher concentration. The guard cells become flaccid and shrink, and the stomatal aperture closes.
Here, there is one activity that we are going to conduct in this process. So that activity we call it as chlorophyll is necessary for the photosynthesis. See here, already we know that at the time of the photosynthesis, plant will absorb the carbon dioxide, sunlight and other water and nutrients from the various parts of the by the help of various parts of plant body, and that is utilized in by the process of chemical process, and the process of photosynthesis will be begins. See here, let us discuss one by one. So each and every component of the requirement photosynthesis components is very essential. See dear children, so for this activity we are going to take as a few two plants here. So take a potted plant having very variegated leaves. That is nothing but the plant that will be in a good condition. So keep it in a dark room for 3 days so that all the starch is used up. So as we know that some of the plants prepare their own food by the help of the photosynthesis, that whatever the prepared food that is utilized in a various metabolic activities that will be held in a plant. Right? No. So but whatever the excess food that is released in a plant body, that is stored in the leaf as in the form of starch, or even that excess food that is released in the plant or manufactured or prepared in the plant, that is released in the form of in the fruits also. So here, when we are going to keep aside for 3 days that plant in a dark room, whatever the excess reserve food is prepared in the plant, that is utilized for their growth and development. Right? No. Then after that, keep it in sunlight for 6 hours. Then the take the leaf. So after the completion of the 3 days, when we are going to keep that as a plant in a dark room right now, so after the completion of 3 days duration, then the same plant only we are going to kept in a when we are going to expose into the sunlight for 6 hours. Then take a leaf from the plant and mark the green areas of the leaf on a sheet of paper. So whatever the greeny areas after the completion of the 6 hours duration, we have to pluck the leaf and we have to mark it as a greeny areas whatever wherever it is present in the leaf, so that we have to mark it, and after that dip that leaf into the boiling water for few minutes. So after the boiling of that wort leaf in a boiling water, again that we have to dip in a alcohol and heat in a water bath. Why we have to dip this leaf into the alcohol? Because whatever the chlorophyll pigments are present in that leaf, that will be dissolves in the oxygen. So then wash the leaf with water and dip into the iodine solution. Then it will be see only the green parts of the leaf that will be turns as a blue black color. This shows that chlorophyll is necessary for photosynthesis. Hope you understand, children. So related to this
An experiment to prove that chlorophyll, the pigment present in the green parts of the plant, is necessary for photosynthesis. So, photosynthesis is an important physiological activity of all green plants. And for photosynthesis to occur, the factors required are sunlight, chlorophyll, carbon dioxide, water, and various minerals and nutrients present in the soil. To prove the necessity of this chlorophyll in photosynthesis, a specific experiment is done in the following way. So, a potted plant with variegated leaves is selected and is kept in a dark room for 3 days. Let me tell you, a variegated leaf is a leaf in which different patches of colors are present, like you can see it in money plant, geranium plant. So the place where green patches are present in the leaf, that portion contains chlorophyll pigment, the green pigment, while the other color patches contain other pigments but not chlorophyll. So we are keeping our experimental plant in a dark room for destarching. Destarching is a step in which the starch already present in the leaves of the plant will be removed. And this step is done to ensure that previously synthesized starch in the leaves get consumed up when the plant is kept in the dark room, because photosynthesis does not occur at that time. So after that, we will again bring the same plant to the sunlight and will keep it in the sunlight for around 6 hours. And now, after 6 hours, we can pluck a leaf from the plant, and this plucked out experimental leaf is immersed in boiling water in the beaker for a few minutes so that the cells of the leaf will be killed, and at the same time metabolic activities of the leaf cells will be stopped, because the leaf cells are killed, so no more chemical reactions will now take place. Now this boiled leaf is taken out from the beaker with the help of forceps, and it is then placed in another beaker containing ethanol or rectified spirit. Then the leaf in ethanol is boiled over a water bath for about 10 minutes. Let me tell you, we are not keeping this beaker containing ethanol directly on the flame, because we know that alcohol is inflammable, it catches fire very easily. So we boil it indirectly. We boil it in a beaker containing water, or we can put it in a water bath. So this is how we boil the leaf. And the boiling of the leaf is done to dissolve the chlorophyll pigment present in the green portions of this leaf. And after boiling, we'll see that the leaf will be decolorized, and then it becomes pale white. Now we have taken this decolorized leaf in a petri dish, and now we will add a few drops of iodine solution over it. It is a starch test where we put an iodine solution to the leaf to test the presence of starch in the different portions of the leaf. Let me tell you that iodine reacts with starch to give a complex dark blue or blue black colored complex. How it happens? Because iodine solution basically is iodine dissolved in aqueous solution of potassium iodide, and this free iodine combines very easily, very rapidly with these iodide to form a negative ion I3, like this. So free iodine reacts with iodide of potassium iodide solution, and it forms I3 ion, and these pink colored balls, a complex of I3 is this. And this I3 complex then reacts with starch to form a bigger dark blue complex, which is appeared here as dark blue color. So whichever portion, green or yellow, will have that starch stored in it, will react with iodine to turn blue black. And we can see here that the green patches have turned to blue black but not the yellow patches. That means the starch was stored in these green patches. That means these green patches were able to do photosynthesis in those 6 hours of sunlight, which proves that chlorophyll is necessary, because the other patches lacked only one factor, and that was chlorophyll, and they were not able to do photosynthesis, and there was no starch, so no turning to any other color, and there was no reaction done even when we put iodine solution. So this is how we test for the presence of starch, and this is how we come to know that how chlorophyll is necessary for to perform photosynthesis.
activity, that is carbon dioxide is necessary for photosynthesis. So here we are going to take as a two potted plants at the same size and keep them in a dark room for 3 days so that all the starch is used up. Then keep the plants in a separate glass plates. Keep a watch glass of some potassium hydroxide on near to the plant to absorb the carbon dioxide. So which means we are going to take as a two power plant potted plants of a same size, and then we are going to keep that two plants in a dark room for 3 days. Why we have to keep these plants into the dark room? Because to usage of starch only, we have to keep the potted plants into the dark room for 3 days duration. Then after the completion of this 3 days duration, we have to separate these two plants and keep a watch glass containing some potassium hydroxide near to the one plant. So cover it. So whatever the two plants that we have taken for the experimentation, for that one plant we have to choose one plant. So for that one plant we have to kept the potassium hydroxide solution for one pot and cover with bell jar and seal the bottom of the jar with Vaseline and make it as airtight, which means after the keeping the potassium hydroxide solution to the one pot of a plant, so ಯಾವುದೋ ಒಂದು ಪಾರ್ಟ್ನ ಚೂಸ್ ಮಾಡಿಕೊಂಡು ಆ ಪಾಟ್ ನಿಯರಲ್ಲಿ ಪೊಟ್ಯಾಷಿಯಮ್ ಹೈಡ್ರಾಕ್ಸೈಡ್ ಸೊಲ್ಯೂಷನನ್ನು ಅಥವಾ ಕ್ರಿಸ್ಟಲ್ಸ್ನ ಒಂದು ಪ್ಲೇಟಲ್ಲಿ ಇಟ್ಟು, ಸೊ ವಿ ಹ್ಯಾವ್ ಟು ಕ್ಲೋಸ್ ದಟ್ ಆ್ಯಂಡ್ completely we have to close that and air doesn't passes through it. So air na ಬೇರೆ ಎಕ್ಸಸ್ ಏರ್ ಔಟ್ ಆಫ್ ವಾಟ್ ಎವರ್ ದ ಅಟ್ಮಾಸ್ಫಿಯರಿಕ್ ಏರ್ ಪ್ರೆಸೆಂಟ್ ಇನ್ ದಟ್, ಸೊ ಅದು ಒಳಗಡೆ ಹೋಗದೇ ಇರೋ ರೀತಿ ಕಂಪ್ಲೀಟಾಗಿ ವಿ ಹ್ಯಾವ್ ಟು ಪ್ಯಾಕ್ ಇಟ್. ಆಫ್ಟರ್ ದಟ್, ಸೊ ಅನದರ್ ಪ್ಲಾಂಟ್ just without the potassium hydroxide solution or potassium hydroxide solution, we have to keep it as a pack as a same just like as what we have done in a potassium hydroxide pot, and both we have to keep this in a sunlight for 3 hours. Then take the leaf from each plant and test for the starch process. So this will be represents the part A, which means whatever the potassium hydroxide crystals we kept in that, completely the starch or the photosynthesis does not takes place because potassium hydroxide absorb the carbon dioxide present whatever it is present in the closed pot, and then without potassium hydroxide pot in that we will be identified the presence of starch, which means because of the photosynthesis will be begins. So these two experiments will be demonstrate the carbon dioxide is necessary for the photosynthesis. Dear children,
dioxide for photosynthesis? Let us verify this by performing an experiment. Requirements: 2 potted plants of same size, glass plate, bell-shaped jar, watch glass, beaker, petri dish, Bunsen burner, forceps, dropper, potassium hydroxide, ethyl alcohol, iodine, water. Procedure: take 2 healthy potted plants which are nearly the same size. Keep them in a dark room for 3 days. Now place each plant on separate glass plates. Place a watch glass containing potassium hydroxide by the side of the plant A. Potassium hydroxide is used to absorb the carbon dioxide. Cover both the plants with separate bell jars. Use Vaseline to seal the bottom of the jars to the glass plates so that the setup is airtight. Keep the plants in sunlight for about 2 hours. Pluck a leaf from each plant. Put the leaves in separate beakers containing ethyl alcohol. Place the beakers in boiling water and heat them. Alcohol will decolorize the leaves by dissolving the chlorophyll present in them. Now, wash the leaves with water. Then put iodine solution on both the leaves. Check the leaves for the presence of starch by iodine test. Do both the leaves show the presence of the same amount of starch? Observation. The leaf from pot A remains unchanged in the presence of iodine. Thus, starch is not present in it. The leaf from pot B turns bluish in the presence of iodine. Thus, starch is present in it. What can you conclude from this activity? Potassium hydroxide placed near the pot A absorbs all the carbon dioxide. Hence, plant A cannot perform photosynthesis. Plant B does normal photosynthesis by utilizing carbon dioxide from the surrounding air.
How does the nutrition will be takes place in animals? See children, it is also very important. So previously this we are discussing about how does the nutrition will be takes place in plants, right? No. So now how does the animal nutrition will be takes place in animals? These are concepts that we are going to now discuss. So let us take one microorganism, best example amoeba. How does the amoeba will in the amoeba how does the nutrition will be held? See, already we know that the amoeba is a unicellular animal, so that we are identified in a living that we will be identified, or this will be survival in a water. See children, how does this nutrition will be takes place in amoeba? See, inside, it will be intake the food by forming the finger-like projections called as pseudopodia. And after the pseudopodia, middle of the amoeba body, it will be forms as a food vacuole. So inside the food vacuole, the food is digested and absorbed, and the undigested food is also sent through out of the body by the help of the pseudopodia only. So that you are going to observe here in the picture. So in the first picture, the amoeba, any food particle that will be come in contact with the amoeba, or amoeba only come in contact with the food particles, so that there is a special modified cell, that is what we call it as pseudopodia. This pseudopodia will be starts increasing its size. So just observe it in between the first and second picture. So the pseudopodia will be starts increasing its size. In this picture also you are going to observe it is a food particle, and then in the second picture that pseudopodia will be starts increasing its size. So in the third picture that you are observing here, the food particle will be entered to the amoeba body, that is we call it as food vacuole. In the food vacuole only, the process of digestion will be begin, and then later that after the completion of the digestion, the digested particles that is completely dissolves in the cytoplasm of the amoeba body. After that, what will be happen? The undigested food will be comes out from the amoeba body by the help of the pseudopodia only. By the help of the pseudopodia only. So this is about the how the nutrition will begins in amoeba. Hope you understand children. So just it is one example that nutrition will be takes place in microorganisms. Hope you understand. Yes.
Nutrition in amoeba. Amoeba eats microscopic plants and animals which float in water. The mode of nutrition is holozoic. In amoeba, nutrition is carried out in 5 different steps, just like the other animals. They are ingestion, digestion, absorption, assimilation, and egestion. Ingestion. When a food particle comes near amoeba, it ingests this food particle by using pseudopodia around it. Digestion. The food is digested in the food vacuole by digestive enzymes. Absorption. The digested food present in the food vacuole is directly absorbed into the cytoplasm of amoeba by diffusion. Assimilation. Food absorbed by amoeba is used to obtain energy for maintaining life processes. And egestion. When a sufficient amount of undigested food collects inside the amoeba, then its cell membrane suddenly ruptures at any place and the undigested food is thrown out of the body of the amoeba. In amoeba, the digestion occurs within the cell. This type of digestion is called intracellular digestion.